Life on Earth is incredibly diverse. It is in forests, oceans, deserts, and even in the small spaces we sometimes ignore. But biodiversity is declining fast. According to the WWF, wildlife populations are, on average, 73% smaller than they were in 1970. This means biodiversity loss is not a future problem. It is already happening right now.
So we took a quick look around and asked ourselves,
what if we could investigate this global issue inside our own school? In our environmental sustainability lessons, we were challenged to see our campus as a place for investigation and action. Our mission was to choose an area and design a scientifically supported intervention to increase biodiversity.
The area we chose is not perfect or untouched. It's close to nature, but it still shows signs of disturbance.
This contrast became our starting point, a human-impacted area besides a powerful natural ecosystem.
The project did not begin with an answer, but with an inquiry.
How biodiverse is this area now? What conditions are limiting life here?
Before acting, we needed to measure biodiversity, so first we used quadrants.
A quadrant is like a sample window.
Because the area is always the same, we can compare the species' abundance, richness, and diversity.
That's how we started a biodiversity living lab. We built the quadrants ourselves doing a maker activity, and then used them in a few to collect scientific data. This also became a moment of community learning. We used the quadrants together and shared how biodiversity can be observed, measured, and discussed. The project became interdisciplinary: environmental sustainability for ecology, math for data, and critical thinking to question evidence. We were not just looking at the area, we were learning how to read it. After analyzing the area, each group received the same mission: to research and propose a biological intervention with one clear goal: to increase biodiversity. Each intervention had to be scientifically supported. So, before building anything, we researched. Only then did we move to action, designing interventions that were not just creative, but also biologically meaningful. Groups designed pollinator gardens, insect hotels, soil biodiversity areas, fungi microhabitats, leguminous plants, and decomposition islands. We investigated, planned, built, and explained our choices. We were not passive students. We were co-creators of a real environmental project.
The goal of this project is to increase the fungi biodiversity in our living lab.
On these holes, we're gonna add rice, and later on, the rice will give good conditions for fungi to grow.
Our intervention project's theme was how can adding organic matter to the soil increase biodiversity. So
we put a soil here to increase the biodiversity of the area by adding organic matter, seeds, and nutrients.
These nutrients are beneficial for decomposers in the soil and for plant growth.
With these nutrients, the plants will grow stronger, eventually increasing the biodiversity.
This attracts other animals for the soil.
We are now adding molded bread to the soil so we can increase the fungi population in our study zone.
We plant these little plants to help the nutrient fixation in the soil. We used this Laboratório system to accelerate the process of growing of our seedlings.
So this is our insect hotel. The goal of our project was to create microhabitats where different species could colonize.
In just a month, our monitoring showed amazing results.
Some quadrants had an increase of up to 73% in the diversity index.
And of course, ecological restoration takes time.
But that's part of the meaning.
Our Living Lab project has no end. It is a long-term space for observation and learning.
It is also in the legacy we leave, a living laboratory where students can study biodiversity, understand human impact, and take responsibility for the environment around them.
We're not only studying the future of biodiversity,
but helping design it.
Whoa!